So students, so far what we have learned is that mitochondria is the energy or the powerhouse for every eukaryotic living cell. Their number varies a lot and they are the largest organelle after the nucleus, and they are the second largest after the chloroplast and nucleus in the case of animal cell and plant cells respectively. We learned in the differences between the two types of membrane. The main criteria was I wanted you to understand that there is semi-autonomous nature of the mitochondria as well as the chloroplast. We'll be learning next. And the second thing was I wanted to explain that how parasites, according to these two scientists, have slowly turned into endosymbionts and helping each other out. So the mitochondria depends upon the nucleus or the eukaryotic nucleus for meeting the requirement of other proteins which it needs, and in return of that it gives ATP to the eukaryotic cell for its needs. Something which I skipped in between was regarding the oxysomes. So what are oxysomes? Oxysomes are also called as the F0 F1 particle. F0 F1 particle. They are also called as the elementary particles, also called as the Racker's particles. So what exactly they are? So these were the structures which were seen by the scientist, and then that's why the name given to it was, you see, chondriosomes, because when high power microscopes, electron microscopes are used, it was discovered that the crista, basically the crista, that means the infoldings of the inner membrane, they carries a large number of racket or lollipop type of structures. These structures are called as oxysomes. So the details tells us that they are 11 nanometer in length, 1.5 nanometer in the breadth, and they are made up of two portions, F0 and F1. Now what is the F0? F0 is this part. It is the base portion, and its function is that it is a proton tunnel. Now tunnel again means it's a transport protein. So it's a protein which is present in the inner membrane which allow the protons to go across and accumulate in this part. So they will go across from matrix into the outer space or the intermembranal space, and when there is a great number of H plus accumulated here, you all know about diffusion and all those topics. When concentration is high on one side and low on the other side, the molecules or the ions, they will try to come back. So as they come back from this F0 particle, they are going to generate enough energy for the F1 particle. Now F1 particle is the head portion, and its name or its function is to synthesize ATP. So ATPase enzyme or ATP synthetase enzyme or ATP synthase enzyme. This enzyme synthesizes ATP, and you all know ATP production is the main function of the mitochondria. So this oxysome is made up of a base. Base is embedded in the inner membrane as a tunnel protein. Then the neck region is just at the surface, and then there is a globular shaped enzyme called as the ATPase enzyme. Head, neck and base. Otherwise the term used is F0 F1 particle. Now let's move on to the next topic, and that is the plastids. So plastids were discovered and named by Haeckel in 1866. Actually, this is the scientist who later introduced the concept of the protista and all. If you remember Monera, protista, fungi, plantae, animalia, so I'm talking about that concept. So plastid word was given by Haeckel in 1866, and plastids are present in plants as well as present in protists, and especially present mention of the name Euglena, because Euglena is an animal type of protist, but it can act like a plant whenever it is exposed to the sunlight. So it is mixotrophic in nutrition, acting like a heterotroph as well as acting like an autotroph. Now plastids, when they originate in the form of a proplastid. So the beginning is a proplastid, a small one. Now it can develop into a leucoplast, it can develop into a chloroplast, it can develop into a chromoplast. So it all depends what is the fate of this proplastid, but it has to become and what is the role it is going to play in the inside the cell of a plant. Now please remember one thing, that the proplastid can give rise to all of these. Even a leucoplast can change into chloroplast, a chloroplast can change into chromoplast, and even a leucoplast can change into the chromoplast, but the opposite is never possible. Can give you some significant examples. For example, when a fruit starts developing, you must have seen fruit develops from ovary. Initially the ovary would be green in color. Green color means chloroplast. And imagine for instance, tomatoes developing. Tomatoes in the beginning are going to be green in color. They slowly change their color into yellowish, and finally they turn into red color. So this change in the color is because the chloroplast is changing into the chromoplast. Opposite is never possible. The proplastids are of these types.